Okay. All right, so we left off here on Monday talking about metaphase. And there's one major event that happens during metaphase, and what is that event?
They line up.
Yeah, the chromosomes line up in the middle of the cell, and the reason why they end up there is because they get to the end of the kinetochore microtubule. A motor protein literally walks them along that kinetochore microtubule until they get to the end. And when they get to the end, the chromosome is in the middle of the cell, and you are in metaphase. And metaphase lasts a very, very short amount of time. Very short amount of time. Of all of the stages, metaphase is probably the shortest stage of mitosis. A lot of cells, when you're looking at cells under a microscope, which you'll do next week for mitosis, a lot of the cells that look like they're in metaphase, if you look carefully, you realize they're not, and they're actually in prometaphase, moving towards the middle of the cell. Metaphase is a very, very short stage of mitosis. So as you look at here, the next phase in mitosis is anaphase. Again, there's a single event taking place in anaphase, and what is it? The two, what you call sister chromatids, that are genetic copies of each other. They are perfect copies of each other, split and start moving towards opposite ends of the cell. Start moving towards these centrosomes that migrated to opposite ends during which stage? Prophase. Okay? Now, what triggers these sister chromatids being pulled apart is the shortening of these kinetochore microtubules. And they start shortening as soon as the chromosome gets to the middle. As soon as the chromosome gets to the end of that kinetochore microtubule and is in the middle of the cell, those kinetochore microtubules start shortening. And that's why metaphase is incredibly short. As soon as they get there to the middle, those microtubules start shortening and pull the two sister chromatids to opposite poles. Okay? So that's what's happening during anaphase, the two sister chromatids being pulled to opposite poles, so that you get the two identical copies being separated into different parts of the cell, so that then you can accomplish the last phase of mitosis. Right? Okay. So let's sum all this up. During prophase, chromosomes condense, the nucleus and nucleolus disappear, the centrosomes migrate to opposite poles and start to lay down those microtubules, the mitotic spindles. During prometaphase, the kinetochore attaches not directly, but indirectly to the chromosome, and they start to move towards the middle of the cell. Yes, sir.
Did you update these slides on Canvas?
Not with the new order.
Okay.
No.
The one you're looking for, that's slide 9.
Yeah. Sorry.
No problem.
Okay. During metaphase, spindle microtubules get to the spindle microtubules, the non-kinetochore, especially function to elongate the cell. The chromosomes move along those kinetochore microtubules, get to the very end, what you call the spindle midpoint or the metaphase plate, and then immediately anaphase starts, and your kinetochore microtubules start to shorten. They pull these two sister chromatids apart to opposite ends of the cell. Yeah.
Okay, so metaphase, both the kinetochore and non-kinetochore do the aligning?
Well, so at the same time that the kinetochore microtubules, so here, let's do this. So these kinetochore microtubules will only reach to the middle of the cell. Okay, that's where they terminate. Okay, so these are kinetochore, and we know they're kinetochore because we'll attach chromosomes to them. Okay, so these are our kinetochore microtubules. They terminate basically right at the midpoint. The non-kinetochore microtubules, and we know that these, wow, that one was a little too long. I got crazy there. We know that these are non-kinetochore because they are not physically attached to a chromosome. They are elongating the cell, and they help to determine where the midpoint is, right? So they're still involved in determining where the midpoint is. The kinetochore microtubules determine where the chromosome stops. But the non-kinetochore are functioning to determine where the middle of that cell is, as the cell is being elongated.
Wait, so how do the non-kinetochore ones not elongate?
How do they not elongate?
Yeah.
Well, they do elongate the cell. What do you mean? How do they not?
In the question it talked about something elongated.
The non-kinetochore elongate. The kinetochore do not.
How don't they elongate? Stupid.
The kinetochore microtubules? No, it's talking about what elongates the cell, not what is elongated in the process.
Gotcha.
Yeah, both microtubules, both types need to grow out from the centrosome.
Mm-hmm.
It's a question of what is actually elongating the cell. So there are motor proteins attached to these that are attached to the chromosomes. There are motor proteins that are attached to these and attached to the other non-kinetochore microtubule. And so as those motor proteins walk this way, they carry this chromosome or this microtubule that way as they walk, and it stretches the cell out.
Is that what remember when you had the guest speaker guy?
Yeah, and he was showing.
Called it walking?
Yes.
Is that this?
Well, that is just a motor protein, and those motor proteins can function in this process. But what he was showing was them carrying a vesicle, like a sac filled with something. But yeah, it's not the same motor protein, but it's a similar motor protein. Yeah, one end, so of every motor protein, one end is mobile and walks, the other end is fixed. So in that one, that was kinesin. The fixed end was holding a vesicle, and the mobile end was walking along the microtubule. The motor protein that functions here is called dynein, and it's fixed on this chromosome, mobile on this one. So you always have one fixed end. That's more than you need to know, but kind of clues you into a little bit of what's going on. Any other questions, thoughts, concerns? Okay, so what haven't we talked about yet of mitosis? Telophase, right? Yeah, we haven't finished it because we haven't talked about the last phase. And the reason why we haven't talked about the last phase is because it happens in conjunction with something that is not part of mitosis. What does it happen in conjunction with? Cytokinesis. Okay, cytokinesis and mitosis are separate events, but telophase happens simultaneously with cytokinesis, okay, which ushers in our next set of questions and then ushers in an opportunity for us to actually work through some of these things. All right, so our last important question that we're going to deal with is how does mitosis relate to cell division, and why is cell division important? How does mitosis relate to cell division, and why is cell division important? You need to remember, I can't stress this enough, you need to remember that cytokinesis is not part of mitosis. So if you're ever asked to explain mitosis, please do not ever say that it is the replication of cells. Okay? It is the replication of nuclei. Okay, so mitosis takes a single nucleus and forms two daughter nuclei. Now, it oftentimes happens in conjunction with cytokinesis, but it doesn't always. Sometimes you can generate more nuclei without generating more cells. Okay? And that's why you have to keep in mind that they're separate processes. All right? We okay with that? All right, so here is telophase and cytokinesis. So during telophase, three main events happen. And what are those three main events?
The membranes, nucleus membranes starts to form.
Yep, the nuclear membrane and the nucleolus reform. They disappeared during prophase. They reformed during telophase. That's one event.
The chromosomes start to unravel.
The chromosomes decondense, right? They condense during prophase. They decondense during telophase. Yeah. Yeah, so that's separate from telophase, but usually that happens at the same time that the cleavage furrow starts to form. The last thing we have to deal with those centrosomes. Okay? And the centrosomes finish resorbing their microtubules. So the centrosome finishes resorbing, bless you, finishes taking apart or disassembling all the microtubules. Now they can be reassembled in the future, in a future mitotic event, or to just build microtubules that are going to anchor organelles in place. But you finish, another way you can say that is you dissolve the mitotic spindle. You dissolve the mitotic spindle. Okay, those are those microtubules growing out of the centrosomes. All right? Cytokinesis, again, remember, it's not part of mitosis, but what's going on during cytokinesis? So as you take a look at this, what's going on during cytokinesis? So we've already split the nucleus into two, right? We finished mitosis. Telophase is either done or is well along the way of rebuilding the nucleus, of decondensing the chromosomes, of dissolving or deteriorating the mitotic spindle. But what happens during cytokinesis? What's that? Yeah, and you noticed this, Josh, right? You talked about we start to form this cleavage furrow. And so now we're going to get a different cytoskeletal element involved. Everything in mitosis is basically governed by microtubules and motor proteins. Cytokinesis is governed by microfilaments. Okay? So you start to form microfilaments along the same equatorial plane, along that same metaphase plate where your chromosomes ended up, and they start to pinch the cell down. Okay? These microfilaments start to pinch the cell down. We talked about cytoskeletal elements a few weeks ago, but why are microfilaments especially well built for doing this? What do microfilaments do?
Do they connect to the plasma membrane?
They do, so that's part of the reason, right? We had different cell junctions. We had adherence junctions that involve microfilaments actually right underneath the plasma membrane. So that's one great advantage of microfilaments. They're associated with the plasma membrane. And they actually anchor a lot of the integral proteins. If they're not supposed to be mobile, they anchor them in place. Okay. Anything else about microfilaments that makes them well suited for pinching the cell in two?
They're small size.
Well, they are small, but remember that the microfilaments are actin. Do you remember talking about that? And then they also associate with a motor protein called myosin, and they form the contraction in muscle cells. Do you remember talking about that? So they're what are associated as myosin, a motor protein, moves along those microfilaments. It shortens the muscle fiber and creates that muscle contraction. So they're well suited to having enormous spaces actually shortening down. And that's what you need if you're going to pinch a cell in two. You need this really big structure, which is microfilaments wrapped around the entire equator to shrink down and pinch the cell into two. Okay? They're great. Really well suited to do that. Yeah.
How when it does that, does it not just like form a hole on it?
Form a hole.
Yeah. How does it like? It's growing back at the same time that it's shrinking it?
Well, the main reason it doesn't form a hole is because the phospholipids won't allow that. Right? Remember, phospholipids self-assemble into a bilayer. Mm-hmm. So if you pinch them closed, they're going to maintain that lipid bilayer that allows the nonpolar tails of the phospholipids to remain free from water influence. So the phospholipids are going to facilitate not just making a hole, of actually keeping everything together.
But if you just like chop it in half, then it would eventually form a hole, but it would like lose stuff.
Right. So, and this is a way you can actually, if you want to, like, if you've ever looked at a cell on a microscope, a lot of times it's a cross section. And so that's exactly what you do. What you do is you freeze the cell or you fix it and then you chop it.
Okay.
Because when you do that, the phospholipids can't work normally. And you can accomplish this if you really cool the phospholipids down. It's like if you tried to get a cell to go through this process of cytokinesis in really cold temperatures, it would probably just cut the cell in half. But it's like if you've ever try to do this the next time you're chewing bubble gum. Blow a big giant bubble and then take like a little rubber band that you use with braces to help, with that, and put it around your bubble. And it'll accomplish the same thing that the cleavage furrow does. If you stretch it out, or a little hair tie too, that'll work. They stretch a little bit better than the braces rubber bands. Stretch it and then put it around your bubble, and it'll do that same thing. It'll create that cleavage furrow, and then it'll, because they're sticky, it'll create you two separate bubbles. You're like, am I going to handle my bubble gum? That sounds gross. All right, so what added challenge do plant cells have in this whole process of mitosis and cytokinesis? The cell wall, right? And so when a plant cell goes through this process, it actually has to build a cell wall along that equator before it can actually split the membrane. So, but it has to happen after mitosis is done or as mitosis is finishing. Right, because it'd get in the way of lining the chromosomes in the center of the cell if you have a cell wall there. All right, so let's sum all this up. How does mitosis relate to cell division, and why is cell division important? The final stage of mitosis, telophase, coincides or happens at the same time as cytokinesis. During telophase, the spindle disassembles. It's one event. Chromosomes decondense. That's the second event. The nucleolus and the nuclear envelope reappear. Those are your three events of telophase. During cytokinesis, a furrow in animal, protists, and fungi, or a cell plate, a new cell wall in plants, forms in the middle and then ends up separating the two cells. And so why is this important? This is the primary way in which a multicellular organism grows. Okay? This is the primary way in which a multicellular organism grows. It's very unusual to have an organism grow by the cells growing larger. Usually a multicellular organism grows and develops as cells divide and you generate more cells. And the nice thing about this is mitosis is asexual, right? It takes the two sister chromatids that are genetically identical and separates them into two different nuclei. So when mitosis and cytokinesis are done, you end up with two cells that are genetically identical, as long as the DNA replication during S phase was done correctly, right? We make about one mistake in every billion or so bases. We've got 3 billion bases in each genome. We've got 2 full genomes. So you'd expect about 6 mistakes every time the cells divide. And this is one of the things, in addition to the telomeres, that actually provides a limit to longevity in a multicellular organism. All right? Any questions, thoughts, concerns, things you feel like you want to share because you want to? All right, so we're going to take ourselves a lecture break. And during the lecture break, I want you to, talking with your groups of 4/5/6/7, I want you to talk about what specific machinery, or at least what those machines would have to do in order to accomplish the different checkpoints. So you've got a G1S checkpoint. What do the machines actually need to do to accomplish that? We've got a G2M. What do the machines actually need to do, or what do they need to accomplish? And then we've got the mitotic, the M checkpoint, or spindle checkpoint, whatever you want to call it. Okay? What actually needs to happen at that point? So how would the cell need to know, how would the cell figure out there's a problem at that G1S transition? How would the cell figure out there's a problem at the G2M transition? How would the cell figure out there's a problem in the middle of mitosis? All right? Take a couple of minutes working with the people in your group, come up with some ideas, and then we'll talk about this together. All right? Take a couple of minutes starting now.
So they would mostly be checking to make sure, do we have enough ATP to copy all? Because that's really what you're doing. Is make sure you have enough energy to copy all the DNA.
In the cells. There's just a DNA to keep itself alive and to
So they're probably like proteins that I would imagine they respond to ATP.
What specific machinery is there, or what it needs to do in order to accomplish what happens after the checkpoints? Oh, there's a lot of different
Protein strength.
yeah, there's like what do you call it? The dependent ones where like after a cell cycle happens, like the product of that cycle stops. Negative feedback. Yeah, the feedback process. That's what it probably is. Like you have a certain protein that's supposed to do something in that phase. And when there's a lot of that protein, then it stops.
Alrighty. Take about another 10 seconds or so. Find somebody in your group to share your thoughts with the class.
Also, make sure that the DNA is properly replicated so we can go back to make sure in the process of checking DNA passes, and then also for the mitosis part checkpoint, you have to make sure that neochores are properly connected to the chromosome. So you have some sort of machinery to check that connection.
So you have a protein that's checking the DNA sequences, and then another protein that's making sure there's
All right. So we're going to start here and we're going to work our way counterclockwise. Do y'all have some thoughts? What do you got for us, Joey?
Okay, so for G1S phase,
Okay.
so that's going from well, it's part of prophase, but it's going from normal cell activity, getting ready to copy the DNA. Probably
It's part of interphase, not
part of
prophase.
interphase, not prophase. Probably something to see how much energy the cell has, to kind of see how healthy it is.
Okay.
So we're thinking maybe a protein that reads like ATP level and stuff, because you're gonna need a lot of ATP if you're gonna copy all.
You sure are. Absolutely. Yeah, it's a very energetically expensive process. Yeah. Good. I like it. Group right here, you have some thoughts to add?
So
Yeah.
the G2M phase,
Uh-huh.
if DNA isn't properly sequenced after that phase, it won't go on to the S phase.
Yeah, so G2, so during S phase, right, we talked about this before, most of the errors that DNA polymerase makes are corrected by DNA polymerase, right? It proofreads as it's writing, but some of them aren't. And then so you have what are called mismatch repair mechanisms, or mismatch repair mechanisms or mismatch repair machines that will go through and will go and fix that. A lot of that happens during G2. And so if by the end of G2, the two copies of the chromosome are not perfect, those sister chromatids are not perfect copies, there's the ability to kind of shut that process down. Yeah. All right. Some thoughts in the back? This is a group of 7, right? I expect a lot from a group of 7.
We were thinking something similar, we'd have to have a spell checker after the chromosome
Okay.
is duplicated. But also, there would have to be something to make sure that the cell is mature enough.
Okay.
Chromosomes.
Right, because G2 is also preparation for mitosis, right? So there are certain things, certain machines that need to be in place in order for that to be the case, right? You need to have enough tubulin to assemble the spindle checkpoint, the mitotic spindle out of, right? You need a lot of materials. So yeah, I mean, one way of just assessing that is chemical maturity. I like it. I like that term. All right, we have some thoughts over here. Is this a group of 4 or is it like a group of 11? Okay. What do you got?
Well, we agreed with everything the previous group said about G1
Uh-huh.
and G2M, but then also we talked about within M specifically,
Uh-huh.
it would need a mechanism to check if the microtubules were connected to the kinetochore correctly.
Yes, and that both chromatids are attached to a microtubule, right? And so that's something that needs to happen. So like what I have here, what I drew for you, and I'm not a great drawer, but it actually worked out nicely. This would be a cell that would fail that checkpoint because the only one of these chromatids is actually attached to a kinetochore microtubule. This chromatid is not attached to this. And because of that lack of attachment, this cell would fail the spindle checkpoint, the mitotic checkpoint, and the cell would kill itself. Apoptosis. All right. We have some thoughts to add, group in the middle. I thought everybody was going to avoid the mitotic checkpoint.
So we said at the G1 S phase, something needs to check to see that the proper growth.
Uh-huh.
And at the G2 M phase, something needs to check to see if there is
Proper.
if it's properly copied
Yep.
in the S phase.
Yep. Yeah.
And then at the M phase, something needs to check if the chromosomes are attached.
Yep, aligned correctly. And this is a wonderful thing, right? G1 is the normal functioning stage of the cell. And so there's plenty of opportunity for the cell to figure out whether or not something's wrong. It's not like it just waits until the very end and was like, hey, did I just screw up for the last, several days? Or have things been functioning normally? Because a cell will kill itself before it gets to that G1S checkpoint if something's wrong. And so G1, even though there's not a specific checkpoint there, a specific machinery, there are specific machinery, not a specific checkpoint, the cell can oftentimes figure out if something's wrong. Yeah, Dylan.
Is it only checking certain things during this checkpoint, or is it checking bundle?
Yes, it's checking a whole suite of different pieces, but it's not checking everything. Like there are things that can get past this point. For example, a virus could have inserted its genome into the genome of the cell, and the cell's not necessarily checking for that, right? I mean, it'll just duplicate it during S phase, and it'll duplicate that viral genome, and this is how viruses can go through what's called the lysogenic phase, where they just kind of hide out in the genome of their host, and their host makes, thousands or hundreds of thousands or even millions of copies of that virus, and then something triggers the virus to come out of the genome and to start killing cells. So yeah, it doesn't check everything, but it does check a suite of different features. Basically, checking everything that it would need to go through S phase and to go through it correctly. So it'll check there for DNA errors, for any big signs that the DNA has been damaged in some way. But of actually just having another genome inserted in, it's okay most of the time. Yeah. We'll go Joey and then we'll go Emily.
Why do cells kill themselves when they find out something's wrong instead of fixing it?
Well, they fix it if they can. But a lot of times it's less energetically costly to kill yourself than it is to actually fix the problem. You have to realize these cells are genetic copies. And so what's really interesting is when you find animals that are very similar. So like think about like a hive of bees or a hive of wasps, where they're not genetically identical to each other, but their relatedness is really, really high. It's energetically favorable for them to just die than it is for them to try to fix whatever the problem is. And so you'll find a lot of bees that will just, they'll just kick them out of the hive. They'll just kick them out of the hive and let them starve to death, or they'll kill them and then they'll eat them, feed them to the larvae.
Okay.
Because you've got another copy. The whole thing is like you have a backup, right? That's what the backup's for. And it's less complicated to just use your backup than it is to try to fix what's wrong.
Okay.
Yeah. That's a good question, though. All right, group in the front.
We just said the same thing about the G1S, how it's going to look over the DNA to make sure it's duplicated.
Okay. Yeah. Well, G1S is prior to moving into S phase, but it's checking for errors. And then the SG2 is, checking to make sure it was done correctly, that the chromatids match. You have two questions?
Yes. Is there something in the G1S checkpoint that checks to make sure that the body can spare that cell while it's going through mitosis?
Well, a lot of that is those are external controls.
Okay.
Yeah, if you're talking about body-wide controls, these are talking about specific checkpoints a cell has to determine if there's an error that needs to just be eliminated, right? I mean, the way you can look at it is it's just a cell's way of telling, is it diseased? And if it is, are you going to fix it or are you just going to eliminate it? And usually the answer is to just eliminate it. Everything, what you're getting at would be external controls. Okay. Yeah, those would be primarily done by contact inhibition and by regulation of growth hormones.
Okay. And then the other one, actually I have two more. Is it possible for a cell to have so many errors that it becomes an issue where there's so many cells killing themselves
Oh, sure. Yeah, absolutely.
Is that
yeah.
like a disease?
I mean, that's what probably causes almost like 90+% of miscarriages would be just catastrophic apoptosis.
Really? Okay.
Yeah.
Then what about neurons?
Neurons?
Yeah, can they go through apoptosis?
Sure.
'Cause that seems like that's if you have a neuron that kills itself.
Yeah.
That's a lot more detrimental. Than like a
Most of the
stomach
most of the problems with neurons is just senescence. Like they just cease to function well, and so they don't transmit signals well. They don't behave well chemically or electrically. But sure, it's possible for a neuron to go through apoptosis. You'll see this happen when neurons become infected with various pathogens. That sometimes they will go through apoptosis.
Is that a big problem for the organism?
Sure. Yeah.
Okay.
Was that? So that's all of them.
Yeah.
Alyssa.
Oh, I was just talking. I thought part of the question was like also the processes by which they go. So I was thinking one of the processes was like negative or positive feedback.
Okay.
Is that how?
Sure. I mean, so your group, Joey specifically mentioned to make sure you have enough ATP, right, to go through that. A lot of that is going to be assessed using negative feedback, right? If you're going through a process that is indicative that you are needing to generate large amounts of ATP, it's a way of the cell determining it probably doesn't have enough energy disposal to go through S phase. Yeah. Yep. Yeah. So the mechanism behind it can be a whole suite of different things, right? There can be a whole suite of mechanisms to let the cell know something's wrong, right? The question is, what does it need to do? What does it need to determine, right? What does it need to check? And then if you wanted to, we could get into the mechanisms of exactly how it checks that. And we'll talk about that in some of them, but not in all of them. All right. So next set of questions. The first one, how is the spindle formed and how does it change throughout mitosis? How does the spindle form and how does it change throughout mitosis? And so here is a zoomed in picture of one of the centrosomes. So remember during prophase, the centrosomes migrate to opposite poles of the cell. And then from this centrosome, you're having microtubules assembled out of this. This centrosome is what's referred to as a microtubule organization center, abbreviated as MTOC. Centrosomes are microtubule organization centers. Microtubules only grow out of microtubule organization centers. But here's what's actually kind of cool. This is bonus information. Microtubules are also microtubule organization centers. So a microtubule can grow out of another microtubule, and so you can get these really branched, complex structures that are really great for anchoring organelles in place and really great for building pathways and roadways. But yeah, centrosomes are microtubule organization centers. So here are the microtubules growing out of the centrosome. And so here is the two pairs of the centrioles within the centrosome. These, we have what are called aster or astral chromosomes, of microtubules. And so as this start to grow out, here's one of these centrosomes needs to migrate over, and that's what's going on during prophase. But as we start to get that, some of these microtubules are already overlapped. They're already overlapped, and they're already connected with a motor protein. And so as this continues to migrate and these continue to get longer and longer and longer, it helps to facilitate the cell growing wider and wider and wider. But the physical contact is made very early on in the process. And so here's, again, continuing to go out through this early spindle where you have some of these microtubules that have been overlapped from the very beginning and are forming part of your non-kinetochore microtubules with motor proteins that, as they're elongating, are also walking them so that they're moving in separate directions and lengthening the cell. And then you can see the nucleus is continuing to dissolve, showing you that we're in prophase, and all of this is going on during prophase. Here's an electron micrograph of the microtubules. And so in here, it's just really showing us the kinetochore microtubules. And how do we know that they're kinetochore microtubules? They're attached, not directly, but they're attached to those chromosomes. Okay, and then those are going to continue to migrate until they reach the end of those kinetochore microtubules. Now, on one side, if it makes contact with a chromosome over here, it's going to have to lengthen this. It's going to have to walk along this kinetochore microtubule as this one is shortened. And it'll go all the way again until the point at which it reaches the end, the physical end of the chromosome. All right. Okay. So let's sum this up, and we'll see if we'll have time to actually get to our next question. So microtubules and motor proteins for the spindle basically are built from these recycled microtubules that are used in the interphase skeleton. So part of that G2M phase is again going to be an assessment. Do you have enough tubulin? Do you have enough material to build your mitotic spindle to actually go through mitosis? So in preparation for this, microtubules fill basically the entire cell, which isn't completely unusual. Microtubules fill most of the cell at the most part, but a lot of the other cytoskeletal elements are recycled and are saved for cytokinesis and other processes. So in most eukaryotes, these microtubules radiate out, forming structures called asters from the centrosomes, which again are microtubule organization centers. So plant cells, they lack centrosomes, but they have other microtubule organization centers. And so there still is a way to facilitate building the mitotic spindle, but without centrosomes. This is a very, very interesting point to know. And this is something we'll come back to on Friday probably, of how it is that you have a group of eukaryotes that share ancestry with all the other eukaryotes, and they lack centrosomes altogether. They use a different microtubule organization center. So during metaphase, where the chromosomes align and actually end up at the middle of the cell, thousands of microtubules compose the kinetochore and the non-kinetochore microtubules. And so if you were to sum up the role of a non-kinetochore microtubule, how would you do it? What is the role of a non-kinetochore microtubule? To elongate the cell. And why is that? What's the point of that? You could cleave it without elongating the cell, right? You could cleave it even if it was small. I mean, you could do this again with what I told you to blow a bubble, right? And that's just going to be roughly spherical, and you can cleave that. But there's another reason why we need to elongate the cell. Yeah.
Is it to keep keep the things inside the cell from like breaking apart or getting like split up, so they can migrate to opposite poles easier?
What things inside the cell?
Like, well, that and also the organelles.
The other organelles? Yeah, there are still organelles present inside the cell.
You're trying to make identical donor cells, right?
Uh-huh.
So you have to elongate it so it's inside.
Yeah, although usually there's some growth that happens immediately following cytokinesis to restore the original size, where you can actually grow Growing a cell is fairly easy. The main way in which you grow a cell is to encourage water to move into the cell, right? And that's going to swell it and grow the cell. The main reason for elongating the cell is to ensure that everything separates properly, right? To give enough space for the chromatids to be built into two completely separate nuclei, and to ensure that all of the organelles are segregated properly. Everything is kept separate. All right? Okay, we don't have time to finish the next question, so I don't really want to get into the next question. We're about 5 minutes before our scheduled end time. So if you want to hang out for those 5 minutes and ask questions, feel free to do so. I will hang out and be ready for those 5 minutes, or otherwise you can venture off and have some extra time to get over to North Campus through the rivers.
All right, hey, everybody, I got to make an announcement here. Okay, so